approach?
Well, first of all, I want to say this N of 1. Let's make sure everyone understands what we're talking about. Historically, the way that therapies have been developed and approved by FDA is by doing a study with people that have that condition against a placebo, and then use various types of statistical evaluation to examine whether that intervention was successful in the outcome against the placebo. I think you're all familiar. This is your standard kind of double-blind placebo randomized control trial. Now, that kind of study is absolutely desirable if you're looking at some class effect, some large-scale effect in a human. But when I say large scale, I'm really talking about a population-based response, a group of people. Now, if I asked you, and this may be a little disquieting or discomforting for some of you, for the major drugs, so let's say the top 20 prescribed drugs in America today, if you had 100 patients treated with a condition that was consistent with the use of that drug, what percentage of those patients would have a positive outcome? Did you get my question? So you have a certain condition, you use the drug that is approved for the treatment of that condition, you take 100 patients and you say, what percentage of those 100 would respond favorably to the drug? It can vary, depending on the drug, between 70 out of 100 to as low as 10 out of 100.
To get approved?
No. Well, yes, to get approved, that's right. So you say, well, how in the world can that be? That means 9 out of 10 people don't respond to the drug, yet it's insurance reimbursed and it's prescribed. It's because when you use a statistical population-based model, you can have some really strong responders that then skew the statistics so that a lot of the people in the curve are non-responders, but they're not weighting much on the outcome variables, these high responders. Now, why is this? Am I saying this today and we didn't say this before? Because you have to know something about genetic variability, which comes post-2000 with the birth of the genetic genome area that we're in now, where we start to see genetic variability at the level we call single nucleotide polymorphisms are far greater than we thought. Our genes are not that variable, but the way that the genes on a single letter alphabets are spelled out in our bodies can vary in the millions from person to person. That's the so-called SNPs, SNPs. So once you start understanding there's much more genetic variability than you thought, and now you put pharmacogenomics on top of that, what happens when you add a drug to and the genes are responsible for metabolizing that drug or having an effect, you see the variation can be very broad, from high responders to low responders. But if you have a lot of high responders, it can push your population study statistics towards positive. Now, how does that then relate to Terry's question of N of 1? So in precision cancer therapy, and you've all been following this major breakthrough in medicine, in which these new drugs are being developed, single or multiple molecules, that actually are uniquely specific for specific genetic mutations in specific tumor types. And when applied to those individuals who have those genetic uniquenesses, the clinical outcomes are unbelievable. They're revolutionary. They're treating these cancers in various forms that were previously very lethal, that now are extending life, sometimes maybe even curing. So precision cancer therapy, then you say, well, how do you study those drugs? Because if you just took a group of people with that diagnosis, let's say melanoma, and you studied that drug that's genetically specific for one form of melanoma, most of the people wouldn't respond. He said, well, that drug doesn't work. The only way you can then define whether that works is to do it patient by patient. That's called N of 1 trials. Each patient is the control against themselves. And this concept was really born out of precision cancer therapy. But now it's been extended because we recognize it's not just cancer. All conditions are unique to the individual. And therefore, what you need to understand is how that person is responding. Now, I could go on in a long discussion, but let me just suffice it to say that we have discounted many, many things that work in some because not everybody responds. I'll say